Cool. Well, take it away.
Hi, everyone. It's been really awesome to be here, and it's a great conference. Thank you. So today I want to talk to you about our efforts towards, the word towards was in some talk in the beginning, building intelligent living machines that have nervous systems. And so far, everyone's talking about brain-machine interfaces, but I'm going to talk about a little bit just machines with brains. So I don't have to motivate this for this crowd why it's cool to build machines. First of all, living machines are pretty awesome. So we've built machines so far out of electronic pieces and wires and wired them up. They're great and amazing, but they also have limitations. Like they can't heal themselves. They can't really adapt to the environment, be autonomous, and also biodegrade when they're done with their work. So it would be cool really to have machines that we can build machines out of cells. And machines with nervous systems would be very useful, because we can have rapid neural signaling. It allows for multisensory integration, all these good things that nervous system does that I don't have to motivate here. And also finally, maybe we can have, with neurons, we could have a substrate for learning and memory. So can we use our conventional engineering approaches for building machines with living cells? And the answer is that it's going to be very, very difficult, because living cells don't follow our rules, and cell collectives also, have different properties that emerge, and their collective properties. So we can't really, take two neurons and imagine a neural network and just try to wire them together. It's just not going to happen. So what we really need to do is we just really need a new paradigm in order to look at, in order to study how do living systems build themselves up. We want to understand the rules of self-assembly and cellular agency and stuff like that, and use that to our advantage when we are building these new machines. So we almost want to ask the question, what would happen if you take a clump of precursor neurons and put them in a completely new motile body? So we want a machine. So we want something that moves around and maybe does some work, delivers a cargo. You can think of applications of machines. What would happen, let's learn, if we put a bunch of neurons in a new motile body? Are these neurons going to develop, differentiate, maybe not, into functional neural networks? And are they going to be able to control body movements? Can they sense the environment? Can they adapt? And if they do that, by what mechanism? And I think this is one of the coolest parts that I'm excited about, because biology is full of hacks. Like all the nervous system has done hacks using the same machinery for doing different tasks. Can they do also in a completely new environment in a developmental time scale as opposed to evolutionary time scale? So these are all questions that I'm super interested in. So to start to answer some of these questions, we start with the motile body part. So this part was the easiest, because this is already a very well established system. It is known that if you take a frog embryo at a certain developmental stage, and you take this top part of it, excise it, and put it in a dish, what it's going to do after half an hour, it's going to heal itself, and it's going to turn into a ball. And after a few days, this ball is just going to move around the dish. So you have your little motile body, and the reason for that is that it is already known that this part of the frog is going to become a tadpole skin. And this tadpole skin just naturally expresses these cells that are called multiciliated cells that have cilia that, works. They beat spontaneously on their own. They have their own little source of energy in their little yolk sac or yolk content. And because of this, these things can move around in the dish. So the question that I asked her was like, okay, so what happens if you put neurons in there? It's just as silly as that. It's nothing complicated, and let's see what it does. So here is an example of one of that top parts, animal caps, that I just stuck a bunch of precursor neurons in. So these are just, undifferentiated neurons that are implanted in this. And then this thing closes up and heals itself again, just like the non-neuron version. And also after a few days, starts moving around the dish. So far, it's viable, it's moving around. And I have a video here. Yeah, so this is one of these neurobots that is moving around the dish. And one of the first things that was clear of just by eye is that these are more elongated and more asymmetrical compared to the ones that don't have neurons. And we showed that this has to do something with the amount of innervation that's inside. So somehow these are the different morphological properties that you can get. And then we want to ask, okay, what's inside? So did the neurons actually grow? So afterwards, you can use for now, some basic immunohistochemistry, but it would be really cool to do expansion microscopy in these. And so here I am staining with an antibody that stains cilia and as well as neurons. And the blue, so that's red, and then the blue are the nuclei of the cells that I actually implanted. And we see that, yeah, so yes, these neurons have differentiated, and you can go through it. So on the inside, they're all neurons that have differentiated, they have their processes, and some of these processes actually go between themselves, but also some of them goes towards the surface of the body that contains these motors and other cells that could control potentially the movement. Now, if you make these out of cells that express GCAMP, you'll see we can get activity, so they're not dead, they're active, and we are in the process of looking at the activity and see how potentially it correlates with their behavior. Another cool thing I want to show you that I like to look at is just the spontaneous patterns of movements that you can get from different ones. So you can get a whole range of various types of spontaneous movement preferences. Some of them like to go round and round in a circle, and you can look at them for hours, and they do the same thing. Some have these cool spirograph patterns. So you can look at that and quantify that, and we've shown that the ones with neurons show a more complex trajectory than the ones without neurons. So maybe neurons are doing something, maybe, we're really, really early at this, so trying to understand. And then finally, we've looked at transcriptomics in these things, and obviously there are genes related to nervous system development, all the major neurotransmitters, receptors, voltage-gated ion channels, everything like that is there. But the part that surprised us was this upregulations of genes that relate to visual processing. So that was really interesting, like the photoreceptors, rods and cones, retinal ganglion cells, all of this machinery is there. So now we are in the process of exploring the stimulus space, looking at stimulus responses, and see what kind of sensory stimuli that you can respond to just naturally without any intervention. And of course, I had some conversations here that's, eventually we can go and say, express particular receptors. But for right now, we are looking at what's just the system does on its own without any kind of intervention. How does it do it? If there is a response, what kind of hack did the system come up with? And then from there, we're going to work on some environmental factors that we can potentially change in the course of their development to kind of nudge them one way or another to generate specific behavioral phenotypes and sensory responses. And finally, we want to learn from the cells themselves how to build living machines that wire themselves up. Thank you very much. Cool. And
wild. The last status update that I had was Xenobots, but
Thank you, that was very cool. If these sort of neurons are, let's say, encoding more complex behavior, what is your intuition for that 0.2 on the mechanism? What do you think might be happening there?
You mean what kind of mechanisms?
Sure, yeah.
I don't know. I don't know. I mean, the thing is that, so it's kind of, for me, it would have to start from behavior, and I kind of sampling the space of responses, what kind of, let's say they respond to light, then it's going to go from there to kind of identify, kind of like the classical way that people have studied animal sensory motor behaviors, which I did for a long time, to understand what's the mechanism.
Yeah, so this is really cool. In the last point, learn how to build intelligent living machines that wire themselves. You could argue an embryo does this, right?
Yeah.
And I'm just trying to get at the nuance of like what you're trying to achieve that's different than sort of a developmental program.
No, we're basically trying to understand how developmental program would operate in novel situations. Because if you want to build machines that do a particular thing, we're not going to stick like a worm in the brain. I don't know. So the idea is that, or we are, but the thing is that you could potentially build these out of human cells from particular person's own cells, and then they would go. And so we want to know what happens in new situations.
You can all volunteer. Okay, thank you so much. That was awesome.